So classification and nomenclature of enzyme में क्या क्या आएगा? Enzymes are divided into 6 classes. तो ऑक्सीडोरिडक्टेज डिहाइड्रोजनेसिस पहला क्लास है, नाम से पता चल रहा है, which catalyze oxidoriduction between two substrates. जैसे एक सबस्ट्रेट रिड्यूस हुआ है एंड एक सबस्ट्रेट, वही सबस्ट्रेट केमिकल रिएक्शन होने के बाद ऑक्सीडाइज हो रहा है. ट्रांसफरेजेस नेक्स्ट है, which catalyze a transfer of a group between a pair of substrate S and S dash. तो आप देख पा रहे हैं इस रिएक्शन में कि G जो ट्रांसफर हो रहा है S से, जब प्रोडक्ट फॉर्म हो रहा है तो S डैश पे G ट्रांसफर हो गया. Hydrolases, enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, CC bond, C halide or PN bonds. Break bond using water. Hydrolases break bond using water. ये important है. Lysis, enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. तो lysis क्या करेंगे? They will leave the double bond and their mechanism would be different than hydrolysis. इसमें भी removal of groups will take place. Isomerases में includes all enzymes catalyzing interconversion of optical, geometric or positional isomers, मतलब cis से trans. तो that would take place by the help of isomerases. Ligases हम DNA application में पढ़ते हैं. Enzymes catalyzing the linking together of two compounds, that is enzymes which catalyze joining of the bonds, for example CO bond, CS bond, CN और PO bonds. So enzyme का एक component होता है cofactor, which is the non-protein constituent bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. ठीक है? तो तीन kinds of cofactors may be identified. First is prosthetic group. That is organic compound tightly bound to the apoenzyme, permanent part of the enzyme structure. तो prosthetic group जो होगा वो permanent part होगा enzyme structure का. Key feature क्या है? Does not leave during the reaction and often part of the active site. Examples would be peroxidase, catalase, prosthetic group which is heme. These enzymes break down hydrogen peroxide, that is water plus oxygen. H2O2 को convert करेंगे water plus oxygen में. So prosthetic group का important part line क्या है? Permanently attached to the organic helper and it does not leave during the reaction. Examples would be peroxidase, catalase. Coenzyme is the organic compound. It is loosely bound and associates temporarily during catalysis. This is different from the prosthetic group. So in important concept क्या है? It comes, participates and leaves. Used by many different enzymes, for example NAD and NADP. So NAD and NADP both contain the vitamin niacin. So the golden line would be coenzyme is a temporary organic shuttle molecule. Next are metal ions. These are inorganic ions and they form coordination bonds with enzymes and the substrate. Examples would be zinc. Which enzyme uses zinc as the metal ion? That is carboxypeptidase. Metal ion helps in substrate binding and catalysis. So metal ions stabilize and activate the active site. So अगर हम tabular form में देखें तो prosthetic group and coenzyme would be organic whereas metal ion would be inorganic. Binding, prosthetic group would be permanent, coenzyme would be temporary and metal ions may be tight or loose. Example of prosthetic group is heme, coenzyme NAD and NADP, metal ion would be zinc ions. Leaves enzymes, prosthetic group would never leave the enzyme, coenzymes can leave the enzyme and metal ions generally don't leave the enzymes. So the role would be structural plus catalytic in the case of prosthetic group. Coenzyme is a carrier molecule and metal ions are for stabilization and catalysis. So follow me for the next videos. Thank you.